Morning. So yesterday you guys started learning about your DNA notes, and that was in this packet here, and you learned about the parts that make up DNA, what it's comprised of, what it does, where it's found. We're going to continue with those notes. So we are on the back of that first page, and we are down to the point where it's talking about, we just finished up some stuff with mutations and things like that. So now we are going to be talking about the process of transcription. So DNA can do a few things. We talked a little bit about replication yesterday, where it's making identical copies of itself, but sometimes DNA needs to be copied in order to make a protein, and that's that process called transcription. In order to go through and make a protein from DNA, we need to make what is known as mRNA, which is known as messenger RNA. So when we talk about some differences between DNA and RNA, RNA is ribonucleic acid. Remember, DNA was deoxyribonucleic acid. So with this ribonucleic acid, there's my R, there's my N, there's my A. One of the differences that we're going to see between RNA and DNA is that RNA has a slightly different sugar. It is called ribose instead of the deoxyribose in DNA. RNA is also a single-stranded molecule, which means that it's smaller. It can get out of the nucleus a lot easier because DNA is double-stranded. It cannot get out of the nucleus. With our RNA, you guys are going to see that there are three types of RNA. There's what is known as messenger RNA that we just mentioned above with transcription. That messenger RNA, you guys will usually see it as that mRNA. We have what is known as transfer RNA, tRNA, and ribosomal RNA, which is your rRNA. Besides having a different sugar, another difference you'll see between RNA and DNA is that in RNA, our nitrogen base pair is going to be replaced. So thymine that we find in DNA is now going to be replaced by uracil, which is that U symbol that you guys see there in RNA. So thymine is replaced by uracil. We also said that the RNA is much smaller than DNA. It's only a single strand, unlike our double-stranded DNA molecule, and it's small enough to leave the nucleus, and with doing that, it's able to carry the message of DNA out to the cytoplasm, which is where my proteins are going to be made. Messenger RNA gets its message from DNA, and that process is called transcription. So we're going to take my double-stranded DNA and we're going to copy a small segment of it and make it into a single-stranded messenger RNA molecule. Once that happens, it's going to go out to the cytoplasm. It gets carried to what is known as a ribosome, and this ribosome is going to be activated or turned on. And that way it's able to kind of read the messenger RNA code to know how to make the protein. So the code on the messenger RNA is now translated at the ribosome by tRNA, and it makes a protein. This process is called protein synthesis. Remember, synthesis just means to make. As you guys are flipping the page, so now you will be going to the next page. The second page in, it's only a single-sided page. If you guys can, just rip that out, and you can actually just toss out the side. It's just a repeat page. So the next page after this one that we just finished with protein synthesis, you should see one that says RNA and protein synthesis, and that's where we're going to start with our next little section. So with our next section, we're starting with RNA and protein synthesis, and a lot of this is kind of what we just mentioned with the last little bit of notes there. So we're looking at the letters RNA, and the letters for RNA stand for ribonucleic acid, and you can see we underline the R, the N, and the A. We know that there are three types: mRNA, which is messenger, tRNA, which is transfer, and rRNA, which is ribosomal. And all three of these are going to help us to make proteins utilizing the DNA message. So why is mRNA called a messenger? And the reason is because messenger RNA gets the genetic code from DNA, and this is always going to take place in the nucleus, that control center of the cell, because that's where DNA is found in every living thing of your eukaryotic organisms. And it carries the code to a ribosome that's going to be located in the cytoplasm. This ribosome is where we are going to make the protein, where we're going to string together molecules in order to make the protein. So down below, you guys are going to see a diagram, and we're going to go through and we're going to kind of label the diagram next to the parts of its descriptions. So mine's a little color coded. If you need to or want to go through and color code this cell, so around the blue is my cell membrane, pause the video, grab some of your colored pencils or markers that you guys may have, and highlight some things if you need to. So we're saying that this entire thing is going to be my cell. Inside my cell, I have my nucleus. We know that inside my nucleus is my double strand, looks like a ladder. So that's going to be my DNA. And off that little segment of DNA, I'm going to make a single strand of messenger RNA. That's going to leave the nucleus, go out into the cytoplasm, find some ribosomes to make the protein for us. So if we're looking at that, and the first part that we have is ribosomes. Ribosomes are the little dots. If you remember your cell flip up that we did, they were the little dots that are located within the cytoplasm, and this is where proteins get made. Proteins are going to be made from amino acids. It's kind of like amino acids are little Legos, and when you put the little Legos together to make that giant Lego house, the protein would be like that giant Lego house. It is made of amino acids. There are 20 different amino acids floating around within your cytoplasm. So the ribosome, which are these dots over here, is the location where we're going to join those amino acids together. Letter A represents the nucleus. And like I said, this is the control center of the cell. This is where we're going to find our DNA. Letter B in the middle we said is our DNA. However, that DNA is going to be making up my chromosome. That chromosome is our genetic material. Usually we'll see it shaped as little X's. Letter C, so we're breaking a part of that chromosome and we're looking at a piece of it. We know that that chromosome is made of DNA. DNA is a twisted ladder or a double helix, so it actually looks like a ladder with little rungs on it. From that piece of DNA, I can make a single strand of messenger RNA, and that's what letter D is representing, my single strand of messenger RNA, and it's going to carry the DNA message, because he put it into his own language, messenger RNA did. He's going to carry it out to the cytoplasm. Remember, this molecule, this DNA, is too large to fit through the nuclear openings or the nuclear pores. Messenger RNA is single-stranded. He can fit through a lot easier. Letter F is all this gooey stuff out here, this liquidy portion. That is going to be our cytoplasm. This is a gel-like fluid environment. Remember, it has a lot of water in it. It also has dissolved particles in it. Letter E is the location where proteins are made. So once again, it's also our ribosomes, because we know our ribosomes are making our proteins. Letter D gets the message from DNA. Letter D, once again, is my messenger RNA right here. He got his message from DNA. Remember, the only difference is here, there is no thymine in messenger RNA, and it's made up of the sugar ribose. So I'll have uracil, adenine, guanine, and cytosine here. No thymine in this messenger RNA. So keep going with the notes. Like we said, if you need to pause that video, go back through, rewind it, look at the portions of it, highlight things if you have to, star your little questions that you may have, and we'll continue with these, and you guys will have a couple little activities coming your way as well. Thank you.